Hello friends, I am Himanshu Jaiswal and today we are going to discuss about one topic, that is protein structure. Proteins are one of the most important biomolecules. They are one pillar out of the four pillars of biological system. The four pillars of biological system are DNA, that is nucleic acid. The second one is the lipids. Third one is the carbohydrates, and fourth are the proteins. So today we will be discussing about the protein structure, how the protein structures, how proteins make some this type of complex structure. We'll see in our presentation. So proteins, they are most important, highly diverse and highly versatile biomolecules. The huge structural diversity of protein enables them, it makes them suitable for performing different types of functions. As you can see in the figure, the proteins can work as hormones, just as insulin. They can work as antibodies, means they can protect us from the microorganisms or pathogens. They can work as the transporter of oxygen. They can work as the enzymes. For example, rubisco, which is one of the most important enzymes in the biological system. This rubisco is the enzyme that combines carbon dioxide and water, and it gets converted into the carbohydrate. So due to this rubisco, life is possible on the earth. The second one is the next example is of ferritin, a protein that binds to iron in your body, and that iron is stored in your body in combination with the ferritin. These proteins can also act as receptors. For example, rhodopsin, which is a receptor of light present in our rod cells of the eye. Proteins also provide contractile forces. They are responsible for the muscle contractions. So these diversified functions of proteins are possible due to their diversified structural features, right? So now we are going to study how these proteins are going to make these complex structures. Now the protein composition. Proteins are heteropolymers of amino acids, right? Most of the proteins are heteropolymers of amino acids. Heteropolymers means they are not made up of same type of amino acids. They are made up of combination of amino acids, right? In biological system, there are 300 types of amino acids, which we can classify into two types. One is standard amino acid and other is non-standard amino acid. The non-standard amino acids are those amino acids which are not going to get incorporated in protein during the protein synthesis, while standard amino acids are those amino acids which are going to get incorporated in the protein during the process of protein synthesis. मतलब जो अमीनो एसिड्स प्रोटीन सिंथेसिस के दौरान प्रोटीन में इनकॉर्पोरेट हो जाएंगे उसे हम कहेंगे स्टैंडर्ड, और जो प्रोटीन में इनकॉर्पोरेट नहीं होंगे, ये किसी दूसरे फंक्शन को परफॉर्म करेंगे, उसे हम कहेंगे नॉन स्टैंडर्ड अमीनो एसिड. For example, PABA, that is para-amino-benzoic acid, is one of the amino acid that has ability to work as neurotransmitter, but it is not going to get incorporated in the protein. So after this, we will study amino acids. We will study how the peptide bonds are formed, and then after we will see the next levels of protein structure. Now amino acids, they are the monomeric units of proteins, right? These amino acids, they are called as amino acids because they have two terminus. One is amino terminus and another is a acidic terminus, that is a carboxylic terminus. This acidic terminus, that is COO negative group, is going to react with the amino group, that will form a peptide bond, and that will release water molecule, resulting in formation of a covalent bond called as peptide bond, or you can also call it as amide bond in the language of chemistry. In the language of biology, we will call it as peptide bond. In the language of chemistry, we are going to call it as amide bond. Now, this peptide bond has some characteristic features. The first and the most important characteristic feature of this peptide bond is that it is having a partial double bond character. Partial double bond character means it is having some amount of double bond character. Around 40% of the double bond character is present in the peptide bond. Now, why it is there? You can see in this figure that this central carbon is associated with two groups. One is the nitrogen and another is oxygen. Both these groups are electronegative, and they are going to shuffle the pi bond between these two bonds, the C double bond O and NH. NH is that C double bond O and CN. So that pi bond will be shuffling between these two positions, and due to this, our peptide bond acquires partial double bond characteristic. If the bond is single, rotation is possible against that bond. अगर कोई bond single है तो आप उसके against rotation कर सकते हो. But if the bond is double, you cannot rotate that bond, and due to this, the peptide bond becomes planar. ये जो peptide bond है ये कैसा हो जाता है? प्लेनर हो जाता है. प्लेनर का मतलब क्या? ये बॉन्ड फ्लैट हो जाता है, इसके अगेंस्ट रोटेशन पॉसिबल नहीं है. As you can see in this picture, due to the planar nature of peptide bond, this is represented as the plate. Around 6 atoms they come in plane, and the peptide bond will not allow rotation around it. Now, which bonds are there which provide rotational ability is the psi bond and the pi bond. The psi bond is it can be rotated by 360 degrees, and the pi bond can be rotated by 360 degrees. But how they will rotate, how much rotation they are going to give, depends on the R chain of that particular amino acid. हर अमीनो एसिड की R chain अलग अलग होती है. जितनी बड़ी R chain होगी उतना ही कम ये bonds rotation देंगे. Right. So now these are the major stabilizing forces for a protein. You can see that there is a disulfide linkage, which is basically a covalent linkage that provides structural stability to the protein. Another is hydrophobic interactions, that means interaction of the proteins with water molecules. Proteins के अंदर दो टाइप के अमीनो एसिड्स होते हैं, बेसिकली 3 टाइप के. Some are hydrophilic, some are hydrophobic. But the hydrophobic amino acids are repelled by water, and they, due to this, the protein conformations are decided. The third one is van der Waals interactions. Van der Waals interactions are weakest interactions. They have very less bond energy, but they contribute high to the protein stability. And the last ones are hydrogen bonding and electrostatic interactions. They provide stability to the proteins because proteins contain huge amount of this interaction. Now, this is a protein. We are going to see all the forces that stabilizes protein in this picture. In the first position, you can see that there is a hydrogen bond which is formed between that OH group and C double bond O group that provides stability to the protein. The hydrogen bonds are, plenty of hydrogen bonds are there, which is written between the two helices, right? The second is hydrophobic interactions between the two hydrophobic groups. They are going to again provide stability to the proteins. The third one is disulfide linkages, which is found between the cysteine residue of the proteins, right? So two cysteines, they are going to join together to form a disulfide bond. And the fourth one, that is electrostatic interaction. Electrostatic interactions are those interactions which occurs between the charged species. जो चार्ज स्पीशीज है उनके बीच में जो इंटरेक्शंस होती है उसे हम इलेक्ट्रोस्टैटिक इंटरेक्शंस कहते हैं, राइट? सो देयर इज अट्रैक्शन बिटवीन द पॉजिटिव NH3 and minus COO minus. So this will provide the stabilizing forces to the protein. Now, there are different structural levels of proteins, starting from the primary structure to the secondary, super secondary, tertiary and quaternary. ये प्रोटीन की स्ट्रक्चरल हायरार्की है. सो इट इज स्टार्टिंग फ्रॉम द प्राइमरी टू सेकेंडरी टू सुपर सेकेंडरी, tertiary and quaternary. So we will see each one in detail. The primary structure is just the sequence of amino acid in a protein. Means if you know the sequence of amino acid in a protein, we know the primary structure of protein, right? And this sequence is ultimately decided by the coding sequence of mRNA, and which is ultimately coming from the DNA, right? So this sequence of amino acid is primary structure. But if you know the primary structure, you can predict the secondary and super secondary and any higher level structure of protein, right? It is possible to means it is possible to predict higher level structure of protein by using the primary structure. Secondary structure. The secondary structures are formed by regular folding of a polypeptide, and this polypeptides are stabilized by the hydrogen bonds. ये जो secondary structure है, ये secondary structure जो होते हैं, ये इनके अंदर regular polypeptide की folding होती है, and these secondary structures are stabilized by the hydrogen bonds. Now, these are some common secondary structures. The first is alpha helix, second is the beta sheets, and the third one is beta terms. We are going to see alpha helix and beta sheets in detail. Now, the alpha helix is basically the coiled peptide which is stabilized by the periodical hydrogen bonds. It is a coiled peptide which is stabilized by the periodical hydrogen bonds, right? Here there are some important properties of the alpha helix which are written here. The first property is the distance between two amino acids in an alpha helix is 1.5 angstrom. Actually, in the straight protein it is 3.5 angstroms. अगर प्रोटीन स्ट्रेट हो तो उसमें दो अमीनो एसिड के बीच में का जो डिस्टेंस होता है वो होता है 3.8 एंगस्ट्रॉम. बट ड्यू टू कॉलिंग, जैसे ही कॉलिंग होती है तो कॉलिंग की वजह से अमीनो एसिड दे come close to each other and the distance becomes around 1.5 angstrom. Next, there are 3.6 amino acids are present per helical turn. एक हेलिकल टर्न लेने के लिए लगभग 3.6 अमीनो एसिड्स लगते हैं. This means that for a rotation of 360 degrees you need 3.6 amino acids. In an alpha helix, each amino acid is rotated by 100 degrees, right? हर अमीनो एसिड कितना रोटेशन देता है 100 डिग्रीज का, और इसीलिए टोटल 360 के रोटेशन के लिए हमें क्या चाहिए 3.6 अमीनो एसिड. नाउ the next one, the hydrogen bonds are formed between nth and n plus 4th amino acid. पहला और पांचवा अमीनो एसिड, nth means first and first plus 4th, that is 5th amino acid, they are going to form hydrogen bonds during the formation of alpha helix. Now there are several proteins which are having high amount of alpha helices. For example, myosin and tropomyosin in muscles, they have high amount of alpha helix. Keratin protein in the hairs and nails, it has high amount of alpha helix. Intermediate filaments of the cytoskeleton, fibrins of the blood clot, they have very high amount of this alpha helical structure. We will see one example in detail. The example is aquaporins. These aquaporins are present on the eukaryotic cell membrane and these aquaporins act as water channels. उनके नाम से ही आपको समझेगा कि ये aqua means water and porins means which provides transporting facility for the water. So these aquaporins, they are present on the eukaryotic cell membrane and they permit the entry of water from one side of to other side, right? These aquaporins are homotetramers, means they are tetrameric proteins, that is they are composed of 4 subunits. Each subunit has a molecular weight of 28 kilodaltons and each subunit is composed of 6 membrane spanning alpha helices. तो हर जो एक सबयूनिट है उसके अंदर 6 मेम्ब्रेन स्पैनिंग अल्फा हेलाइसिस है और ऐसे टोटल 4 सबयूनिट्स मिलकर के एक एक्वापोरिन बनता है. मतलब एक एक्वापोरिन के अंदर टोटल अराउंड 24 अल्फा हेलिक्स प्रेजेंट होते हैं. एंड ईच सबयूनिट ऑफ दिस एक्वापोरिन एक्ट एज एन इंडिपेंडेंट वाटर चैनल. ये ध्यान रखें कि ये जो एक्वापोरिन है इसका हर सबयूनिट इंडिपेंडेंट वाटर चैनल के जैसे एक्ट करता है. नाउ the second secondary structures are beta sheets. Beta sheets, they are formed by the aligning of zigzag shaped peptides, right? So जो peptides होती हैं they adopt zigzag shape and one peptide is present after another peptide and when they are aligned together they form the beta sheets, right? So beta sheets are formed by multiple beta strands. जो इंडिविजुअल आप देख रहे हो उसको आप बीटा स्ट्रैंड कह सकते हो और ऐसे बीटा स्ट्रैंड की जो अरेंजमेंट होती है उससे हम बनाते हैं बीटा शीट. द डिस्टेंस बिटवीन 2 अमीनो एसिड्स इन द बीटा शीट इज अराउंड 3.5 एंगस्ट्रॉम व्हिच इज 3.8 एंगस्ट्रॉम इन केस ऑफ स्ट्रेट चेन प्रोटीन. नाउ we'll see there are 2 types of the beta sheets. One is parallel beta sheet and another is anti-parallel beta sheets. Parallel beta sheets are those which are having same N terminus towards one end and C terminus towards other end. मतलब दो पेप्टाइड्स हैं जो N से C टर्मिनस की तरफ रन कर रही है, तो पहली पेप्टाइड का N टर्मिनस जिधर साइड होगा उधर ही दूसरे पेप्टाइड का भी N टर्मिनस होगा, और ऐसी पेप्टाइड्स को हम बोलेंगे पैरेलल बीटा शीट. While the next type is anti-parallel beta sheet, where the N terminus of one peptide faces C terminus of the other peptide, right? But the parallel beta sheets are comparatively less stable as compared to the anti-parallel beta sheets. Now, what is the reason behind that? आप फिगर में देख सकते हो, you can see in this figure that in anti-parallel beta sheets the hydrogen bonds are linear. There are straight hydrogen bonds, but in case of parallel beta sheets the hydrogen bonds are not collinear. Due to the linearity of hydrogen bonds, the beta sheets are anti-parallel beta sheets are more stable as compared to the parallel beta sheets. Now, one structure which is formed by the combination of multiple beta sheets is called as beta barrel. This beta barrel is a component of porine proteins. These porine proteins are present on the bacterial cell membrane. They are also present on the mitochondrial membrane. And these porine proteins act as non-specific transporters. They can transport any molecule which is having size less than 600 Daltons. Right? So herein you can see this. There is a beta barrel structure which is passing the membrane. The exterior side is there, the interior side is there, and between that there is a membrane, and it is spanned by this beta barrel. Now this beta barrel will act as a transporter for various types of molecules. Now we'll come to the super secondary structure of proteins. Super secondary structures are those structures which are formed by combination of more than one type of secondary structure. ये ऐसे structures हैं जो एक से ज्यादा सेकेंडरी स्ट्रक्चर के कॉम्बिनेशन से बनते हैं. यू कैन सी इन द फिगर दैट देयर इज अ जिंक फिंगर मोटिव which is having one alpha helix. It is also having a beta sheet. It is also having two beta sheets which are stabilized by the central zinc ion which is shown in the figure in green color. Right. The second example is the leucine zippers. They are also called as basic zippers. These zippers are they are present in the DNA binding motif. जो डीएनए को बाइंड करते हैं वहां पर ज्यादातर जगहों पर ल्यूसिन जिपर ये मोटिव्स होती है. दैट इज दे आर गोइंग टू रिकॉग्नाइज पर्टिकुलर सीक्वेंस ऑफ डीएनए. राइट. नाउ वी विल गो फॉर द टर्शियरी स्ट्रक्चर. टर्शियरी स्ट्रक्चर इज द थ्री डायमेंशनल स्ट्रक्चर ऑफ प्रोटीन, एंड हाउ द प्रोटीन adopts टर्शियरी स्ट्रक्चर will depend on the primary structure, that is the sequence of amino acids present in that. Right. But one major force that also decides the tertiary structure is the hydrophobic interaction. During the formation of tertiary structure, water repels hydrophobic amino acids in the core of protein. And hydrophobic moieties are turned towards the water, and due to this the protein three-dimensional structure of protein is adopted. These proteins are going to change their structure when you are going to transfer it from water medium to any other medium which is non-polar. Right. So the tertiary structures are basically the three-dimensional structure of proteins that a protein adopts when it is incorporated in water. Right. So this is the tertiary structure of myoglobin. Right. And we'll go for the quaternary structure. Quaternary structures are formed by the combination of more than one tertiary structure. So quaternary structures are formed. They are formed by more than a combination of more than one tertiary structure. Each tertiary structure can be called as a subunit. So this quaternary structure can be called as multi-subunit, right? Because it is having more than one subunit. It is having more than one tertiary structure. So you can see in this figure there is the structure of hemoglobin, which is composed of four tertiary structures or four subunits. You can say that there are two alpha chains and there are two beta chains. Now, how these structures come close to each other and how they associate. These structures can be associated with each other with the help of various types of bonding which are listed. First is covalent bonds, that is disulfide bonds which are present between the cysteine residues. Next is the hydrogen bonding, electrostatic interaction, hydrophobic interaction, and van der Waals interaction. all these interactions will work for the formation of the quaternary structure. Thank you.